So to tell us a bit more about forensic DNA evidence, could you please give a warm welcome to the Professor of Genetics from the University of Leicester, Professor Turi King. Thank you for joining us. And as you know, we're going to be talking about DNA. You have a great history in DNA. Why don't you tell us a little bit about your background?
So I got asked to be involved in a really amazing case. So I led the genetic analysis which proved that the remains of a skeleton found under a car park in Leicester was that of King Richard III.
Oh, that was such an incredible case. And so I'd like you to talk to us about DNA.
I will
Floor
do that.
is yours.
Okay. The DNA in our cells is in nice long strands that contains our genetic code. So this is the double helix. This is what our DNA looks like. Now what I'm going to do is I'm going to show you some DNA. And the way I'm going to do that is actually with some spit. This is my spit, so it's a little bit gross. So what I need to do is I need to break down the cell membranes so the DNA can come out of the cells and into solution. The way I'm going to do that is by adding some washing up liquid, just a few drops.
Just ordinary standard washing up liquid.
I'm going to give this a swirl because what I want to do is I want to get those cell membranes breaking down so we can release the DNA into solution. Now the next thing I'm going to do, we're going to add some salt solution. So all this is just salt dissolved in water. And what the salt does is it helps the DNA strands stick together. And then what I'm going to do is I'm going to add some really cold alcohol. And what the alcohol does is it precipitates the DNA out of solution because the DNA can't stay in solution when there's alcohol. Where those two layers meet, you're going to start to see the DNA, and it looks kind of like clouds. And those are the little strands of DNA. There you go. So that's DNA just there. Now, how do we use DNA for a crime scene? So what we do is we produce what's known as a DNA profile. And I've actually got one here. And it's not the entirety of somebody's DNA, which is known as a genome. What we're doing is it's like we're taking little snapshots of little sections of our DNA that we know are really variable between individuals.
So if we were to think about, we've got some DNA samples that are taken from the crime scene. Do we have a DNA database?
We have a national DNA database. It's not everybody's DNA on it. It's DNA from people who have committed a crime. They're the ones who have got their DNA
Nobody
on the database.
in this room then?
No.
Absolutely not.
No.
So when we were looking at fingerprints, we were able to get a match between some fingerprints in our scene and some fingerprints on the fingerprint database.
Right.
How would we go about doing that similar comparison for DNA?
Well, we need a DNA profile. Now you can think of your DNA as being a bit like a book. So our DNA is made up of 4 different building blocks, A, C, T, and G. And we are looking at what's known as DNA markers. So bits of our DNA where we know we differ between individuals. And you guys are going to be our national DNA database. That's why you've got these booklets. So the markers that we look at are like a stutter in the DNA. They're known as an STR, a short tandem repeat. So in our little booklet, we have got 23 pairs of chromosomes. 1 chromosome 1 has come from your mom, and 1 chromosome 1 has come from your dad. And that's what we've got here. Now then, let's have a look for one of these stutters. What I want you to do is I want you to go down to line 7, and I want you to look for the word cat. And I want you to count how many times it's repeated. They have to be next to each other all in a row. If you see a cat on its own, not interested. I want you to do the same for the right-hand side. Find cat and count it up. Cool, okay, so that's 1 marker. You've got 2 numbers. And we've done that for 6 markers. And they're all done in your book. I want to see if there's a match to the profile that I've got here. So, do any of you have a 10 for either of your chromosome 1s? If you do, give yourselves a little mark like that. If you've got a 5 for either of your chromosome 1s, give yourself a mark. Okay, next one. Chromosome 2, check your chromosome 2s. If you've got a 10, give yourself a mark. If you've got a 9, give yourself a mark. Guys, ready for chromosome 3? If you have got a 3, give yourself a mark. If you've got a 6, give yourself a mark. So you're either going to have 0, 1, or 2 marks. And finally, chromosome 6. If you've got a 7, give yourself a mark. If you've got an 8, give yourself a mark. So now what I want you to do is I want you all to stand up. How many of you has got 0 matches? Okay, if you've got 0, sit down. Sit down if you've got just 1 mark. How many of you got 2? Sit down if you've got 2. Ooh, wow, that's going quick. 5, sit down. 6. Ooh, who's got 6s? So if you've got 6 matches, that means you could be a parent or a child or a sibling of the perpetrator. How many's got 7, 8, 9, 10, 11? I think you've got 12. Okay, if you've got 12 matches, that means you are a perfect match to our DNA profile. And so it's not you who's the perpetrator, but you are holding the sample that is a perfect DNA match. So it looks like we've got a match to our crime scene in our DNA database. And that's really amazing because we've already got a match as well, haven't we, for our fingerprints?
We have a match for the fingerprints, and
Yeah.
it looks now we might have a match for the DNA. And it's not always perfect, is it, in
No.
terms of a match? Sometimes samples get degraded.
That's
there
right.
are issues. We don't always get a perfect match. And that's why we go to the experts then for their opinion.
Exactly.
So what else is available to us now in terms of DNA, perhaps that we didn't have before?
Yeah, so what you can do now is, if you want to use DNA and you're looking for matches, is you could take DNA from a crime scene, look at a lot of different markers, and upload it to a genetic genealogy database. And what that does is it's not just looking for a perfect match. You can get matches with somebody who's like an aunt or an uncle or a cousin or a second cousin. And that allows you to build family trees and allows law enforcement to start to hone in on who the perpetrator might be. Now, it's used in various countries around the world. It's not used here in the UK yet. So these days, people are using DNA to try and do things like predict what a criminal would look like. It's called predictive DNA phenotyping.
Whoa. So that's taking the DNA that you had in that little layer, and it's saying, if I analyze those little clouds in that little layer,
Yeah.
I can predict what somebody looks like. That sounds like science fiction.
That's what they're trying to do. So they're looking at genes that we know code for things like hair color, eye color. People are trying to look for genes to do with face shape. And they're even starting to analyze DNA where they hopefully are going to be able to say how old somebody was.
Thank you very much, Nita. I find it rather scary, I have to say, but isn't that the wonderful thing about forensic science? This is just science pushing the next step forward.
It is.
Thierry, thank you so much
You're
indeed
welcome.
for taking us through DNA. Thank you.
Really